Hello everyone and welcome back in the third day of the training program, which is about the RNA pull-down procedure to identify RNA targets of a non-coding RNA. So just a quick reminder for what we cover in the first and the second day of our training program. So we talk about the RNA pull-down assay, actually the first day or the first part of the assay, and then we talk about the long non-coding RNA, and we gave a brief background about this kind of RNA, and then we talk about the hybridization step. And what left is we need to cover the oligonucleotide probe, how we design antisense DNA oligonucleotide probes that is specific to long non-coding RNA of interest. And what also left is the second day or the second part of the RNA pull-down assay. So to design the antisense DNA oligonucleotide probe that's specific to long non-coding RNA of interest, we can use the primaries. We have first to generate the secondary structure. So we can use the primary sequence of the long non-coding RNA of interest, and then we generate the secondary structure on the RNA structure web server. And actually there's so many of websites we can use to generate the secondary structure for the RNA. So RNA structure web server or Vienna RNA web server, and also I will talk about other servers that we can use for this purpose. And here are some examples of the sequence of the probes, DNA oligonucleotide probes. They're just some names of the probes. So basically we have a few notes before we start designing the probes. So first we using the primary, as I've mentioned before, using the primary sequence of the long non-coding of interest, we can generate a secondary structure using whatever web server. So basically on these web servers, we can find different algorithms that can be used, and there are so many tools. There are actually 3 prediction tools that can give the best result for the probe design. Those are the RNA fold that we're going to show you today, and max expect, the highly probable base pairs, and the probe not probable base pair, including pseudonodes. These analyses can be performed and compared. Other web servers such as Vienna RNA WebSuite 7 also can be used. But we have to pay attention to the following things. First, we have to select the region that display low probability of internal base pairing and design antisense oligonucleotide probe of 25 bases with a strong affinity for these regions. And we have to note that the GC content of these probes should be compromised between 40 and 60% using alignment search tool BLAST. We will show you how we can manage this percentage. We have to make sure that the selected antisense oligonucleotide probes don't recognize nucleotide sequences in other RNA expressed in the chosen cell system. Second, we need to design also a non-specific DNA oligonucleotide probe of 25 bases, which display neither affinity for the long non-coding RNA of interest nor for other RNA sequences in the genome of interest. And we can order the probes with the biotin at the third end. So we need to label the probes. Note, we have here a note: in order to reduce the steric hindrance, the distance between oligonucleotide and the biotin, the label, has to be increased with the triethylene glycerol spacer for an optimal result. And to be able to assess the specificity of the result of pull down, it is recommended to design 3 different antisense oligonucleotide probes and then to experimentally compare their efficiency. So basically, we go through a protocol, the web server. Okay, so we have a few notes before we go to the server. So basically, as we understand, we need first to generate our secondary structure based on the primary sequence of the long encoding RNA. So to do that, we have to go to open web browser and enter uh, this link, and then paste or upload our RNA sequence in the corresponding text box. Okay, and then we click proceed and wait till the process complete. The graphical model and dot brackets notation result of the RNA secondary structure will appear. So basically, I mentioned there's three prediction tools that can give the best result for the probe design. One of them was the RNA Fold web server. So we're going to use this for our training program for today to generate the secondary structure of the primary sequence of the long non-coding RNA. So basically, this is how it looks like the the page for the RNA Fold web server. So basically, we need here to put the sequence, the primary sequence, and we need to check these things, that the minimum free energy and operate and partition function we have to check on, and avoid the isolated base pairs we have also to check it, and interactive RNA secondary structure plot. They can show you the plot, RNA secondary structure plot with reliability annotation and mountain of plots. So basically, you have to check all these and then put our primary sequence of the non-coding RNA of interest, or any RNA of interest in general. So basically, in the next slide here, we are showing we are just copying and pasting the sequence of the RNA of interest in the right place, and then we hit proceed. Basically, as soon as calculations are done, we can access our results on this website, but I will go through this all the steps in details, because we need to adjust a lot of things, but this is in general. So basically, when the results come out, you can see this we can see the sequences are here, the secondary, and we can this is without any highlighting, without any coloring, the base pairing. We can choose one of these, no coloring, color by base pairing probability. Okay, we can play with these options, and we can see here the graphical output. Okay, because we chose to show up the structure and the graphical output. And here, if we chose to, if we hit the color positions, we will see the sequences in all in colored and showing the base pairing. And we can choose no coloring. So the purpose here, we can see the base pairing. So after we generate the secondary RNA structure based on the primary sequence of the long non-coding RNA, the next step we mentioned previously that we need to design the non-specific DNA oligonucleotide probe of 25 base pairs, which display neither affinity to the long non-coding RNA of interest nor the other RNA sequences in the genome of interest. So it's better to give an idea or background, a brief background about the probes. So basically, we'll give introduction about the probes. So basically, the probe is a nucleic acid molecule, single-strand DNA or RNA, with a strong affinity with specific targets. Probe and target-based sequences must be complementary to each other, but depending on the condition, they don't necessarily have to be exactly complementary. Genome probes are used in various blotting and in situ techniques for the detection of nucleic acid sequences. These oligonucleotide probes are synthesized chemically as oligonucleotide based on the information available on the amino acid sequence of the protein of interest. It can be used as a probe in the identification of gene which encode for that particular protein. Oligonucleotide probes are generally targeted to specific sequences within genes. The most common oligonucleotide probes contain 18 to 30 bases, but current synthesizers allow efficient synthesis of probes containing at least 100 bases. The selection of oligonucleotide probe sequences can be done manually from a known gene sequence using these guidelines. The first one of the guideline, number 1, the probe length should be between 18 and 50 bases. Longer probe will result in longer hybridization times and low synthesis yield. Shorter probe will lack specificity. Number 2, the base composition should be 40 to 60 GC, 40 to 60% GC. Non-specific hybridization may increase for GC ratio outside of this range. Number 3, be certain that no complementary regions within the probe are present. These may result in the formation of a hairpin structure that will inhibit hybridization to targets. Number 4, avoid sequencing containing long stretches, more than 4 of a single base. Number 5, once a sequence meeting the above criteria or the previously mentioned criteria has been identified, computerized sequence analysis is highly recommended. The probe sequence should be compared with the sequence region or genome from which it was derived, as well as to reverse the complements of the region if homologies to non-target region greater than 70% or 8 or more bases in a row are found, that probe sequence should not be used. So before we go to the website for designing the probe, we have also a few notes. So basically we need to provide the exact target sequence, not only the probe sequence, we also need the target. So for designing we need the target sequence, and the probe will target the sequence that we will submit. And we need to know also that we need the cDNA RNA sequence of the target, not only the protein sequence. This is just a replication for this note. So basically this is the home page for the program that we're going to use, the DNA software application portal. So it will look like this. And we go to the projects, okay, and we choose a new project, okay. And after that we're going to choose the model that we are using. In this case they are showing the zebra fish, okay, the fish, not a human. So after we choosing the RNA source, which is the zebra fish RNA, we will this page will show up. Next we will have, so we'll show up so many target sequences. We have to pick one of these and enter the accession number. So we have to enter the accession number and choose one of these sequencing targets. So here showing in this slide the place where we write down the accession number, and then we click next. So here we can choose the type of the format. So in our case we want to design antisense DNA probe, and then we can choose the length range. How long is it? So people sometimes 20 to 30, they do 20 to 30, sometimes it's 24 to 24, the same number is fine. Then we hit next. So on this page I'm showing that we can use the same length, exact length. So 24, the maximum and the minimum will be the same. So it's either the same or difference. Here we can, so after that we will have this result will show up, the sequences, okay? So basically we can target any sequence, okay? So so in here, if we want to target, we just enter the target range, which nucleotide to which nucleotide we want to target. Like if we want to because in our procedure we need to like design it so it has like little affinity for or no affinity for specific sequences, so we need to highlight these, so the probe that we are designing not targeting specific sequences. So next we can set up the hybridization condition using and by entering these information like annealing temperatures 37, the probe concentration, the target concentration, okay, and we can choose the type of the probe, so what we're going to put at the end, which I mean the type of the labeling, so what we're going to put at the 5' end. So in this case they're showing FAM, fluorescein, but in our case we're going to put the biotin, because it's the probe should be labeled with biotin at the 3 ends, so we have to change the location, okay, and the label, and then we hit next. And after that we also can adjust the content of the content of the GC content of this probe. We can compromise it, and as previously mentioned, it should be between 40 and 60%. So we can use these alignments search tool. So here's 50 is fine, it's within the range. And after that we can see our result, and by clicking on view results. So we will have so many results showing up, okay. Basically we will choose the one with the high sensitivity. So this is a sensitivity, so we have 97, 98, so this one should be the best to be used. So by reaching this slide, we reach the end of the day 3 of our training program, and tomorrow we'll be talking about the last part of the RNA pull-down assay, which is about the RNA isolation step. Thank you.